Hello everyone, a very warm welcome to all of you. Today we are going to introduce you to stable isotope resolved metabolomics analysis. In today's webinar, you will learn the essentials of peak picking on metabolomics data processing tool, Almaven. You will also understand the stable isotope resolved metabolomics workflow and how to analyze the data within minutes on our platform, Poly. Before we start, please note that this webinar is being recorded and a copy of the recording will be shared with you after the webinar. We will take all the questions in the last 10 minutes of the webinar. We have a support team, so if you have any questions during the demo, you can post them on the chat and they will reply promptly. So I am Surbhi Podar. I lead the development of our product suite for metabolomic researchers at Elucidata. We have with us today Dr. Pawel Lorkiewicz. Dr. Lorkiewicz is the director of the mass spectrometry core facility at the University of Louisville, an integral part of Diabetes and Obesity Center. Today, he will introduce SIRM data analysis to us, followed by a live demo on a dataset generated in his lab. Before I hand over the webinar to Dr. Lorkiewicz, I would briefly like to introduce you to Elucidata and its capabilities. Elucidata is a 60-member based company based in Cambridge and New Delhi, with a deep expertise in building pipelines and tools for analysis and interpretation of integrated omics data. We've been part of 20 publications, and some of you may have also been at some conferences like ASMS and CPSA where we presented recently. Uh, Our key product offerings is our platform Poly, which is a customizable platform for comprehensive analysis of integrated biomedical data. Poly is a cloud-based platform and is highly customizable for data processing and interpretation. It also supports end-to-end processing of metabolomic data. Poly integrates data from a range of tools, including Almaven, Thermo Fisher's Compound Discoverer, and Symphony from Waters Corporation to support a range of downstream analysis like flux analysis, absolute quantification, and many more such. Our current products also include Almaven, the metabolomics data processing engine that simplifies speed picking in your data with an automation model. Almaven is open to Windows, Mac, and Linux users and comes with an intuitive user interface to facilitate effective peak integration and peak picking. So you can read the book chapter on Almaven published this year in Methods in Molecular Biology by Springer Nature. As a platform, Poly solves the important challenges associated with processing, interpretation, and collaboration of large data sets. We realized these are some of the critical challenges that are faced by metabolomic core facilities. This brings me to our latest product, a comprehensive mass spec omics offering for core facilities. Poly's customizability ensures seamless integration with existing analytical workflows to provide maximum value to researchers at your core facility. Poly users at core facilities now share their data with interactive reports on the cloud. More importantly, we ensure that core facilities choose the products that are specific to their immediate needs. Our dedicated team of experts also identifies the best solutions that we can build to suit the user needs. Specifically, Poly is completely vendor agnostic, so we are compatible with all major instruments and analysis, including MS and MSMS data from LC and GC columns for all mass spec experiments. Our core facility offering is already increasing data throughput at Yale University, the University of Louisville, and VIB. We are also partners with Waters, SciEx, and Bruker to break down, that actually helps us break down barriers in metabolomic researches. With that, I would like to hand over the session to Pawel.
Thank you so much for the introduction, Kirby. My name is Pawel Lorkiewicz, and I will talk about stable isotope metabolomics and how we use L-Maven and Pawel workflow with our data. I will be able to perform the demonstration and show how we can get from Let me start again. I hope now I'm audible. Um, my name is Pawel Lorkiewicz, and I will talk about stable isotope resolved metabolomics and present a demonstration of the L-Maven and Poly workflow with the data that we acquired and show you um, how we process the data and how we can get from raw output to actual analyzed data in minutes. So without further ado, let me talk a little bit more about what stable isotope resolved metabolomics, or SIRM, is. SIRM, as the name implies, is a type of metabolomics that uses stable isotope labeled tracers or substrates to follow fates of individual atoms into product metabolites. Below you will see a comparison of the sort of classic metabolomics approach and the SIRM approach, just to give you an idea of the features that you get from both. In classic metabolomics approach, we typically measure abundances of metabolites, so snapshot concentrations between experimental groups. While we can get a lot of information from that, certain types of information is not readily achievable. Specifically, it's difficult to pinpoint to a specific pathway, especially if the metabolite can be produced through multiple pathways. And sometimes concentrations may actually be steady due to homeostatic effects or compensation effects. So we may actually not be able to see any differences between groups, while actually there are changes, metabolic changes within those groups. Therefore, we have limited knowledge of the pathways involved and of the pathway activity or flux. With SIRM approach, we are using stable isotope-labeled tracers, and those actually result in enrichment with the label in downstream metabolites from that particular substrate. That allows us to pinpoint on specific pathways and determine relative activity or fluxes of those pathways. We can actually get a better idea of the contribution of nutrients to specific metabolite pools. And just like in classic metabolomics, we still get that pool information or metabolite abundances. Just to explain a few basic concepts in SIRM, as I mentioned, SIRM uses stable isotope-labeled tracers. Stable isotopes are actually non-radioactive forms of an element. They just have more neutrons than the non-heavy form of the atom. And stable isotope tracers, because those stable isotopes actually occur in nature, they're non-toxic. So unlike radioactive forms, they're safe to the organisms and can be widely used. Therefore, in vitro and in vivo experiments are possible. Because there is a difference in the mass of neutron, those labeled metabolites can be easily detected and differentiated using mass spectrometry, and therefore mass spectrometry is a widely used analytical technique in that type of approach. If you look at the bottom left corner, we have an example of commonly used stable isotope tracers. This is a fully labeled C13 glucose, but other tracers like fully labeled C13 glutamine, fully labeled C13 fatty acids, pyruvate, lactate, and other metabolites are frequently used in stable isotope tracing experiments. The way an experiment is typically performed is that tracer is introduced either in vitro into cell culture or given in vivo as a nutrient included in the diet or in an infusion in vivo experiments. There have been experiments with animals as well as in humans. And the plot that you see here is a typical enrichment plot, and I will talk about it a little later, but just to kind of give you an idea of what happens in SIRM experiments versus a non-labeled experiment or classical metabolomics experiment. The black bar shows you sort of the non-labeled alanine, and as you can see, the majority of that alanine is unlabeled, and that there is some contribution of C13 in there, and that comes from natural abundance. Whereas in a classical stable isotope tracing experiment, you will see enrichment with heavy atoms, and thanks to mass spectrometry, you can actually tell apart the number of heavy atoms that were incorporated into that alanine or any other metabolite that's downstream of the tracer. And you notice that there is a sort of an uneven enrichment pattern and different amounts. And those different numbers of heavy atoms that were incorporated into a molecule, they're called isotopologues. So those are different forms of the molecule with different numbers of heavy atoms. So moving on, just to describe a general workflow and an expertise required in a typical SIRM experiment. Those experiments are not easy, as many of you may know. They require typically a team of experts, and there are different stages to each of those experiments. At the beginning, just the design and execution of an experiment requires a lot of biological expertise and the knowledge of stable isotope tracing, as well as communication with analysts and frequently statisticians and bioinformatics specialists. That stage involves the choice of the proper tracer, depending on the pathways of interest or the scientific question. It also requires planning appropriate controls, many of which are not labeled, using the appropriate pharmacological controls, and establishing the right time of the experiment to reach the metabolic and isotopic steady state. And once the experiment is performed, which takes a lot of time and effort to plan it properly, we have to deal with the enormous amount of data that is generated from such experiments, since labeling results frequently in very complex data sets. And when we talk about platform considerations, of course, we will consider, well, in this example, in this demonstration, I'll be talking about mass spectrometry. It produces very complex labeled data sets. Once this data is acquired, that data has to be properly analyzed. And this is one of the biggest headaches of SIRM analysis, because the data needs to be deconvoluted, assigned, natural isotope abundance needs to be corrected for, and fractional enrichment and pool data needs to be properly ascertained. These processes are not easy, and I will show you the general workflow in a moment, but typically this has been the biggest bottleneck that was taking days, weeks, or sometimes months to deal with. And of course, once the data is assigned, then the part of interpreting that data, plotting and displaying it properly and interpreting it is the final stage. As I said, it requires multiple expertise and a communication between various scientists. So today I will demonstrate data analysis from the experiment that we recently performed. We used pharmacological controls to sort of establish define metabolic configurations. We kind of knew what to expect, and therefore we could predict certain enrichment patterns and establish state controls, metabolic states. The knowledge of that can actually help in designing future experiments and using those as sort of reference controls. So we use koningic acid, a known inhibitor of GAP, as well as we used FCCP, which increases the flux through TCA cycle and stimulates mitochondrial oxygen consumption. So those are sort of two polar opposites, and we expect very pronounced differences in those two experiments. And the way we designed the experiment, we obtained neonatal rat cardiomyocytes. We grew them in the medium containing an excess of uniformly labeled C13 glucose with or without the pharmacological controls. We grew that for 12 hours. Then we quenched the metabolism, extracted it with 60% acetonitrile, and subjected it to LC-MS analysis on thermal Q-exactive Orbitrap with UHPLC system. After the separation, we acquired full mass spectra, and that pretty much was used for analysis. Before I actually go through the demonstration, I'll just quickly tell you about the typical SIRM analysis workflow. I'm sure many of you are familiar with those. Depending on the platform that is used, that process is what takes a long time, from hours to days to weeks. And from my personal experience, I started out performing such analysis with multiple pieces of software with very poor user GUI, and that would take weeks to months. Now with the help of Maven and Poly, I can actually, our lab can do it in hours, which is a huge increase in throughput, and really reduces a lot of data analysis related headaches. But let's get to the workflow itself. Once the data is obtained, we have to perform spectral deconvolution and assignment of metabolites. Then after we've got we've got that step done. And this is also, just to kind of give you an idea, this is a difficult step because we're not dealing with just an unlabeled peak, but we also have to assign all the isotopic peaks depending on the number of carbons or labeled atoms. We can deal with anywhere from 5 to sometimes even 40 carbons or other heavy labeled peaks to assign. After that is performed, we need to make sure that we remove natural abundance. And here you have an example plot of labeled citrate. Natural abundance correction is important and really cannot be done manually because if we don't perform this step, the interpretation of data may be flawed. And here, if you look at the isotope log n+6, you will see that after natural abundance removal, we almost completely lost this isotope log at n+6. And that, if it disappeared after natural abundance correction, we may actually change our interpretation completely. So the final step after that is performed is typically two plots are generated. One is of the fractional enrichment, and the other one is just for pool, total pool information, which basically sums up all the areas for every isotope log peak. And with that, I will go ahead and show you how I use the LMaven and Poly workflow. Let me just switch to my computer real quick. So we start our analysis with the LMaven, which is software that is available for our desktops, and this is a pretty straightforward interface. It has two major components. The first component shows you the samples, and I've already preloaded all of my samples. So we had 3 groups, each one containing 3 biological, not technical replicates. And as you can see, we have the control FCCP treated group as well as the koningic acid treated one. Then, of course, you have the chromatogram, and then in the compounds tab, we have to load a metabolite list that we will use for assignments. We generated that metabolite list simply by running standards or unlabeled samples and using our Metabolomics, classic Metabolomics setup. We established which metabolites we can confidently detect. We validated that. We included molecular formulae and retention times. That is a part of the database that is basically loaded. Those are all, the database is just a simple comma delimited file. Samples, however, are basically any mass spectrometer output that can be converted to NZML format. This is something to stress is that LMaven software as well as Poly, those are platform agnostic or vendor agnostic. And while I'm working here with thermal Orbitrap data, Poly can work with Waters, can work with Bruker and other vendor files. So that's a very big advantage, especially for core labs or labs that that have different vendor spectrometers, and they don't really have to worry about it working or not working with their spectra. So once I've loaded all my files and uploaded the a list of metabolites, I can perform assignment, and all I have to do is just click on peaks, and I will work with default data just to show you how LMaven does this. In this window, first of all, notice that there are multiple options that can be changed, but the default options actually work very well. We select our database, select the M over Z window. Of course, the larger the window, the more we will assign, but the more likelihood of misassignments and catching peaks that do not belong to the metabolite. We can match the retention time if we have that, and the number of groups per compound basically means how many assignments do we want to include. Of course, because this is labeled data and LMaven can do both labeled and unlabeled data, we have to choose the port isotope peaks right here. And we just need to make sure that we detect the proper isotopes. So in our case, it's C13. And once we've done that, then all we have to do is click on Find Peaks. The assignments are performed very quickly. And here, just to quickly take you through all the assignments, I will just explain all the windows here in our interface. So top left corner, I'm not sure if you can see my cursor here, but basically the list that's in green, it just shows you all the assignments, all the hits within the database. So sometimes if something is not in green, it means that it was not detected, but it is in the database. So this is just an example database. We detected everything. And all the detected metabolites are listed in the peak table on the bottom right corner right here. Again, I'm not sure if you can see my cursor. I hope you can. In the spectrum window, you will see essentially whenever I click on the metabolite, you will see the proposed assignment. It is important for us to actually go through those assignments because LMaven, as good as it is at assigning things, it doesn't always make the appropriate assignment. And we can see it here in case of the 2PG, it just found some noise peaks and assigned them as such. This red dotted line, it tells us pretty much where the retention time expected for the metabolite is. So that kind of helps us actually locate the proper assignment. And now that I clicked on it, on the bottom left corner, you will see an isotope plot. This is a very good feature of LMaven. It actually shows you fractional enrichment. This is not natural abundance corrected, but for every sample, you can actually immediately see the enrichment. So this is kind of a way of vetting your data very quickly in the interface. Notice that one of the samples here is missing, and if you look up, you have the peak area top, right? So this essentially lists all the peak areas for each sample. And again, you see that the sample K25 is missing this assignment. And that's because we're using real data. This actually happens. This is not a very abundant metabolite. However, LMaven is very good at peak picking, and we can go really low with our signals. So I just have to double click on this, and it opens a bookmark table that essentially has this new assignment, and I can delete the old assignment. So I'm just going to quickly check all of my assignments in the peak table just to make sure that they were picked properly. And as you can see, it it found most of the peaks, and just clicking on each one of them, you see that there's a very specific isotope pattern in each group. So this visual representation helps a lot just by going through data and making sure that all the assignments have been performed properly. And this really doesn't take a long time, but is very helpful. And we can quickly learn about some issues that may arise. And we can also correct misassignments on the fly. So I'm just going to go through this list. The list is not long, so bear with me. Notice that this interface is very user-friendly. All the colors are very helpful. Okay, so here's another one, pyruvate. Again, we have to change it to a different assignment and just delete the old one. Again, everything is done very quickly, just using intuitive shortcuts or buttons. And once we're finished with that, which we are, so far I have all the assignments made, and it took me just a few minutes, including essentially going through each assignment and correcting them. Once this is done, I just need to include my corrected assignments in the peak table. And once this is done, I am ready to submit this for analysis using Poly. So Poly is the online companion to LMaven. LMaven basically takes you through spectral deconvolution and peak assignments. Once everything is assigned, we can immediately submit this data online to Poly, and there's just a button on the top left corner that says Poly. And once we click on that, it logs us into our account, and that may take 30 seconds to a minute, depending on the speed of our connection. And that's it. So we have 3 applications to choose. We will go with PolyPhi. This one, this particular one is made for stable isotope experiments. We select the table, so we only have one peak table. We also select the groups. Groups basically mean LMaven has a scoring system. If you take a look at this peak table, there are a lot of different values here that are displayed, and one of them is sort of the score, and LMaven scores peaks as good or bad. Currently, I'm just going to go with all groups, create a new project. Let's just call it 13 demo, and upload my data. This data can also be exported as a CSV file, as you can see in this peak table view, and can further be looked at in Excel. Actually, if someone wants to do some manual overview of the data, this is a very good feature, and you can certainly do that. So once the data has been uploaded, we just click Start Fluxing, and what happens is it opens the browser window and loads our data. I don't have my email application open, but we actually receive an email once this is uploaded to Poly, and that email contains a link that we can always revisit. So that's a very good feature, because we can go back to this data and re-edit it again at our convenience. So this opens an Excel spreadsheet and that's in Google Sheets. And this lets us define our cohorts. So I'm just going to quickly assign some abbreviations for our cohorts. Okay. Oh boy, I'm having some problems with the sheets. Let me quickly check something. Okay, let's try again. Sometimes Google creates issues for me. Well, we may actually start with this email and try to recreate it again, because that's exactly what happened. Let me try and open it again. So I'm just going to try and reconnect. Okay, let's see if it lets me do it now. The issue actually here is with Google. Sorry about that. I'm hoping that this will solve the problem. Okay, here we go. Yes. So once we enter the cohort information, the actual process happens. So as you can see, this is really hands-off at the moment, and it takes us through the entire workflow automatically. And this is probably the biggest advantage of this software, that it is all contained in one. So there are no modules that you have to go back and forth. After the files are validated, the natural abundance is corrected automatically. And after this is removed, the fractional enrichment are also calculated and the data is merged. Those can be actually done manually within the Poly interface, but this is a very convenient way of doing this, since basically everything has been combined into one. So this is pretty much it. All the final step is just generating visualizations. And as you can see, this entire process takes a few minutes. From my personal experience, this process used to take me days to weeks sometimes. So the improvement is just incredible, and it's really tremendous in terms of the information and data throughput. Once the visualization has been created, we have another window in Poly. And as you can see, I'll start from the top and just kind of explain everything, and then just take you sort of on a, just to show you what this output is and how we can take advantage of it and what we can do with it. So just the very first thing on the top, you see that there are sort of 3 steps in this workflow, and that's a very important one. It makes it so it is not kind of a black box. So you can always go back to your quality check, or you can go back to your peak picking, and actually, if there's anything that was misassigned, that you believe that that data has not been properly vetted, you can always go back and re-edit that data and remove metabolites that are bad, or the groups that are marked as bad, as you can see. So as you can see, there's going a little lower on your right side. That's literally what it says. You can filter those metabolites by sort of good or bad groups. I will not do it. I'll actually show you. I like to work with real data, and that's what I'm showing you here, is that there are situations where you see large error bars because maybe the metabolite was not detected in one of the groups. Then underneath, you have a list of all the metabolites that are detected and processed. Below that, you have the isotopologs for each metabolites that were detected and that are plotted. The 2 plots that you see here are the fractional enrichment plot and pool total plots. Note that you can download them, and you see that little icon of PNG. If you click on that, that automatically downloads it for you. And really, you can just open it and use it in your publication, in your PowerPoint presentation. So this is already processed data and visualized and pretty much ready to interpret. So as you can see, we went from raw data to processed data in a matter of minutes. So the second one is the pool total, as I was telling you. So that essentially is telling you about the classical metabolomics metabolite concentration. This takes into consideration all your isotopologs, so that bunches up together. And I'm just going to kind of show you how we can interpret that data. I hope this will work. I'm going to split the screen into 2 just to get some pathways for our reference. So I'm just going to start with glycolysis pathway and just kind of take you through the metabolites that we detected and just show you what kind of information we can get. I will not interpret everything. Of course, we don't have time for that. But I'm just going to show you specific patterns that we can see that help us really get an idea that the data was actually properly assigned, and that this is aligned with sort of not only analytical expectations, but also with biological expectations. So starting with glycolysis, we start with fully labeled glucose, that's 6 carbons. And if we look at glucose 6-phosphate that is downstream, and notice that we have 3 major isotopologs here. And we have the M+6 isotopologs, which means that all 6 carbons have been labeled. There's an M+3, and there's M+0. So that's telling us that essentially that fully labeled glucose was incorporated, and most of the downstream G6P pool was actually labeled. There is some contribution of unlabeled, of course, from intrinsic sources, and that we see as an n+0. If you look at the pool total, you will see that the koningic acid treated group had the highest pool total for this G6D metabolite, since it kind of blocks glycolysis at the GAP. We would expect it to be higher than the other groups. Now going down, let's move to 3-carbon metabolite, the GAP. And we see that once we click on any of those metabolites, everything refreshes almost on the fly. Please note that in the FCCP group, we have a large standard deviation, large error, and that's due to the fact that one of the metabolites was missing. And I'm just going to quickly show it to you here. Going back to L-Maven, you see that there's something wrong in this particular FCCP14 sample, and that pretty much shows here in our plot. Other than that, we see a similar behavior. The pool, the metabolite pool in koningic acid treated group is much higher than the others. So we can go down that pathway, look at our 2PG group, and things just sort of change in terms of the pool total. But we see kind of the continuation of labeling that's expected. It goes from 6 carbons to 3 carbons, and almost the entire pool is labeled with the stable isotope. So now switching back to another pathway, Krebs cycle intermediates. I'm not sure if I can make it any bigger for you to see. I hope this is big enough to kind of get an idea of these metabolites. Let's see. Let's just start with pyruvate. So this is also as expected. We have pyruvate labeled at 3, and we also have some contribution of unlabeled pyruvate. So that makes sense. Again, we go from 6 carbons in glucose down to 3 labeled carbons, and of course there is some intrinsic non-labeled source pool of pyruvate here too. Moving along the cycle, let's take a look at the citrate. So here we have a much more complex pattern, but this is a very important and very interesting pattern here. So looking at the fractional enrichment plot, now we can really tell that we have used sort of pharmacological controls, and that those metabolic states are completely different. So with the green being control, if you look at the yellow group, which is koningic acid, there's hardly any labeling in citrate. So we have 80% of unlabeled citrate in koningic acid group. Means that there's very little labeled glucose getting into that cycle. Well, FCCP is pretty much fully labeled with maybe like 5% of unlabeled citrate, and what you see is that really, as we sort of expect from the way FCCP works, we actually see that that enrichment at n+6 and those higher isotopologues is higher, is more pronounced. There is not very much happening in terms of the pool totals. So if you think about it, if we just performed a classic metabolomic experiment, pool totals are not changing significantly, but there's a huge there are huge differences happening sort of under the hood, if you will. And I'm just going to show a few of the other Krebs cycle intermediates just to give you an idea. So let's go to fumarate, and you see there's hardly any difference in the labeling pattern. Again, a similar behavior, but let's do malate, for instance. It's practically the same. So, this is giving a pretty good indication in terms of the data integrity. If you look at error bars, they're very small, meaning that we have a very reproducible data set, and that essentially the software does a very good, stable job of processing those spectra, and really every step is working very well. So the final thing I want to show is just kind of how we can track the metabolism and get some idea of what's happening. And let's take a look at the purine metabolism. I just hope I can make it any bigger. Okay, so looking at the purine metabolism, and I will take the AMP as an example. So you see that there's an isotope-labeled at N+5, and if we look at the metabolism here, the fully labeled glucose going through the pentose phosphate pathway results in 5 carbons of ribose, and we can actually see that right here. Interestingly enough, that fully labeled glucose can go through multiple pathways, go to fully labeled serine, and result in 3 carbons in the purine ring. And we also see that especially in the FCCP group. So this is just kind of showing how we can really get a lot of information about the pathway activity, pathway involvement, just from the labeling patterns, and how much we can tell just looking at the differences at those fractional enrichments in our data set. So just to kind of conclude this, I will show you what else we can get out of this interface. There is a way to download the data set, and we can download it as a comma delimited file and further open it in Excel. And that actually helps because we can display the data in different ways and process it in the not just in Excel, but for example, our group uses Prism to plot such data, and with the help of Elucidata, this process can also be automated, and Elucidata is very helpful in terms of writing scripts to streamline certain processes and customize them. So that also really helps save time with data analysis. So I'm going to finish here and conclude this talk. I just hope I could show you how the LMaven and Poly workflow is helpful in terms of saving time and gaining through analysis throughput, and helps really with the stable isotope resolved metabolomics or similar data sets that use stable isotope tracers. How flexible and easy the interface is, and how much it actually can save us time and headaches related with dealing with such complex data sets. And with that, I will conclude this demonstration, and I will be happy to answer any questions. So I'm looking at the question. Okay, so Prasad asked, what's the reason of using 60% acetonitrile? So 60%, we have a very specific sample prep protocol, and typically what we do is we extract metabolites using acetonitrile, water, and chloroform, and this is a sort of a three-layer established extraction protocol. The chloroform extracts nonpolar fraction of metabolites, which is typically lipids, phospholipids, and acetonitrile and water has been found to be very effective in extracting polar metabolites. So essentially 60% ACN is giving us the best yield and extraction efficiency for polar metabolites, and that's kind of how we established our LCMS workflow too, and it works the best. I hope that answers the question. Now, the second question is, how useful is SIRM in untargeted metabolomics? That's kind of a loaded question. I would say we use untargeted in metabolomics. So let me just kind of describe the workflow that we have, and maybe that will kind of answer the question. We actually start with an untargeted metabolomics approach, and we don't use SIRM. It's basically easier in terms of establishing the metabolites that we can detect, and establishing a list or a database of our metabolites. And we use untargeted metabolomics with no labeling to get the list or to get the library. And once we have a list of those targets, then we use the SIRM approach with Almaven, as I showed, simply because it helps us, and that's a targeted approach, right? So we kind of do the untargeted to targeted approach for SIRM. Now, one could do SIRM in the untargeted fashion. The problem is, if you end up with a very complex labeling pattern, you don't assign just the parent peak, which typically for untargeted approaches, that's actually stripped from natural abundance. So you're looking at your parent unlabeled, the M0 peak. Here you would have to have multiple labeled peaks used for your untargeted. So I would say it's not a very efficient approach, and I would not recommend it. I see another question. Can you separate G6P and F6P in this case? What was the column used? Okay, truth of the matter is, we cannot really separate G6P and F6P. I would say that G6P, I just used one of the labels, but truth of the matter is it should be hexose 6P. We cannot really separate those isomers very well. So things like 2PG and 3PG is also difficult for us to separate, and G6P and F6P. Just for the purpose of this demonstration, I just used one of the labels, but in our publications, we actually use hexose 6P. As far as the columns go, we have a pseudo 2D LC approach. We use reversed faced column, and we also use a Hillig column. For For the reversed column, we use the waters HSST3, and for, sorry, let me turn off this software. So for the reversed column, like I said, it's a waters HSST3 column, and for the helic column, I believe we use one of the millipore zig helic columns. And the way we perform the analysis, we actually we inject our sample on both columns at the same time, and the separation is happening in parallel. And then we collect whatever comes out of these columns, and we detect sort of the sum of the two. That increases our coverage and also helps us pick the more intense signal for our metabolites. We have, if you look my name up in Google Scholar or PubMed, there should be a few publications that have that method, but I will be happy to also email those for you. Okay, let me move on to the next question. Isotopologues carries variable number of radio-labeled atoms. How does this help to determine the exact mechanism? So first of all, we do not do any radio labeling. So as I said, stable isotopes are not radioisotopes. So that's the first thing. But I think that's just a nomenclature. How does that help to determine the exact mechanism? So as I showed the example, we can pinpoint a very specific enrichment pattern, fractional enrichment pattern. Like I showed with AMP, many of the isotopologues can be very easily assigned to a particular pathway. So for instance, if you have 5 C13 carbons in ribose, that is indicative of involvement of pentose phosphate pathway from uniformly labeled glucose. Therefore, if you see the M+5 isotopologue in purines and pyrimidines too, you can actually know with good confidence that you are looking at that particular pathway. Similarly, if you go through glycolysis, if you see fully labeled, say, GAP, that means that basically it came from fully labeled C13 glucose. So that the fractional enrichment and the pattern and the number of isotopologue really pinpoints to particular pathways. Of course, this is simplifying things, but it is quite accurate with many mechanisms. And we use controls, pharmacological controls, to further confirm those. There are also other techniques that confirm the labeling patterns, and the MARA is used very commonly to do so. I hope that answers this question. I think I have another question coming, and that's from Amar. As illustrated in pyridine mechanism, number of carbons appearing in sugar bases constituted from earlier derived isotopologues, which I may see was 1 carbon in every single step, leading to 5 total labeled. But if it is variably labeled, how it may account for? I am not 100% sure if I understand your question, but I'll try to, let me see if I can share my screen again and just show you what I'm talking about. So let us take a look at this particular mechanism. So if we start with fully labeled glucose that has all 6 carbons labeled, when it gets transformed or metabolized to C13 labeled ribose, that process basically removes 1 carbon and you end up with fully labeled ribose. Therefore, you will see 5 carbons. The ribose enriched with all 5 carbons. The likelihood of having just 1 carbon is very low because you would have to really take that glucose and pass it through multiple pathways, and that would result in scrambling of these pathways and involvement of any other ancillary pathways. They're very unlikely to happen. Very typically, pentose phosphate pathway is very fast. It's energetically favored, so the likelihood of having just 1 or 2 carbons instead of all 5 is very low. Similarly, starting with fully labeled glucose and moving all the way to serine results in fully labeled serine. I think I may actually have serine in my assignment, so I can actually show you. Let me see if I can pull serine up. Okay, there we go. There is serine right here. I'm not sure if it will be labeled. Yeah, so you can see here that serine right here is labeled at M+3 or M+0. So going back to our AMP, we can actually see that right here at M+3. Notice that there are other isotopologues, and that's true. There is labeling coming from other pathways, but the contribution can still be inferred. There are multiple publications on that. In fact, there is a very good paper from Hunter Mosley that models labeling of UDP glucanac. Again, I will be happy to send it after this talk, but at this moment, I don't really have time to dwell into all the mechanisms. Of course, that's a longer discussion. Okay, do we have any other questions? Can you find, okay, there's another question from Marksu. Can you find new metabolites beyond your known pathways with the setup? Yes, absolutely, you can. So the biggest advantage of labeling, and again, that's a question of whether we want to do untargeted approaches or targeted approaches. But either way, so the answer is kind of twofold. The first one is, yes, you can find new metabolites, and if you see labeling of those metabolites, you can definitely find not only new metabolites, but you can also find new pathways. And again, there are quite a few recently published publications, papers that show, and that's specifically in cancer, but we also have a publication in review right now in Scientific Reports that's showing involvement of pathways that were not expected or that were not previously considered, especially in cancer metabolism or cardiac cell metabolism. So the fact that the metabolites are labeled, that's also very helpful, because if you inspect the spectrum, the mass spectrum, and you notice that there is labeling in a particular metabolite, that indicates that metabolite had to be synthesized, and the carbons from your tracer or any other heavy atoms were involved in, were contributed to that particular metabolite pool. So even if you have an unknown metabolite or an unknown peak, if you see labeling in that particular analyte, that helps you kind of ascertain that that's a metabolite. Whether it's new or if it's just something that's not considered at the moment in targeted metabolism, that's a different story. But essentially, that helps you find new species through their labeling pattern. I hope that makes sense. So I'm not sure if we have time for any more questions. All right, there are no more questions as I can see. And with that, I will conclude my talk. I hope this was helpful, and I hope that I convinced you that LMAVEN and Poly is worth our time, especially in those type of data analyses, and that it actually saves tremendous amounts of time. And with that, I wanted to thank you everyone in this seminar, and again, thank you and bye.
For this amazing webinar, and thank you all the attendees for being such an interactive audience. We really enjoyed running this demo webinar with you. If you have any questions, you can reach out to us on polydemos@elucidata.io. We'll be happy to answer all your queries there. So thanks.